Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I am your host Dylan Hamilton, and today we're going to talk about GMOs, otherwise known as genetically modified organisms, and this is our continuation of A22 biology, guys, and let's just get straight into it. I'm not going to waste any time. So, we start off by GMs, which are just genetically engineered modifications, or microorganisms, sorry. Genetically engineered microorganisms, these are more or less the same thing as a genetically modified organism. So, a chemical is produced by a GEM, a genetically engineered microorganism. It's usually insulin. We'll use insulin as an example as we go through it. So, bacteria will produce insulin, which is identical to the hormone that is naturally produced by humans. Previously, insulin was extracted from dead domestic animals, but this was time-consuming and ineffective. So now we use a different way, because non-human insulin is different to the human insulin, which can cause allergic reactions. So other products include the human growth hormone, enzymes, adhesives, surfactant protein, and interferon. So we're going to go through how we actually make these GMOs now and how it actually works. So transgenic plant is an example. So a soil bacterium will cause the growth of tumor-like plant galls. Bacteria can enter a plant where it is damaged, and this can integrate the bacterial DNA into the DNA of the actual plant, and this can cause rapid cell growth and the formation of these plant galls. The gall-forming genes are then removed and replaced with donor DNA. We use things called gene guns, which are used to insert the donor DNA into host cells. So we coat these microscopic pellets with DNA, and they are fired into the host cell, and hit or miss donor DNA is actually becoming incorporated into the host DNA. So it's less reliable when you use agrobacterium, which is used with species that are resistant to infection by the bacterium. Most transgenic plants are called genetically modified crops, or GM crops. So what are the benefits of having these GM crops then that have these donor DNA inserted into them? Well, you have herbicide resistance, so it's a more effective application without the need to avoid spraying near the crops. It also has pesticide properties, so the bacterium will produce a protein that is toxic to insect pests, which reduces losses to insect pests in terms of the plant. It also reduces the use of insecticides, so less expenses, and there's also less harm caused to non-insect pests, and there's also less pollution. Disease resistance, because they're resistant to fungal and viral pathogens. There's a greater ecological range because plant varieties are produced more in the and they're more drought resistant. And finally, nutritional enhancement, because they have an improved nutritional composition and content of their goal. So there's increased protein content, texture, and flavor. These are developed to produce compounds that are beneficial to man. Examples include vaccines and drugs. There are arguments against having GM crops. And the first one is that they're not natural. The second is that you can have the creation of superweeds, because whenever you transfer genes, cross-pollination can occur, which means you have to carry out a rigorous risk assessment and make sure safe precautions are taken to ensure the risk is low. GM crops have a greater ecological range. We said this was an advantage. It can also be a disadvantage, because GM crops can grow in a wider range of habitats and outcompete these non-crop species, reducing biodiversity. Some insects can become resistant to the toxic effects of the GM crops with this Bt gene, they call it. And finally, they worry that some compounds produced by GM crops could actually cause allergies themselves as well. So, transgenic animals, and we've talked about plants. What about animals? Well, we include the encapsulating donor DNA in a lipid vesicle called a liposome, and they use a lipid carrier, which is easier for the DNA to cross the lipid bilayer. Another method is electroporation, which involves disrupting the cell membrane by making it more permeable by the use of high voltage treatment. Viruses will insert the gene causing a respiratory disease, and this is altered genetically so the host cell will not be destroyed. And this is useful for delivery of genes to patients. We use microinjection, which injects the DNA directly into the nucleus of a fertilized egg as well. Thanks for watching this video. I've been Dylan Hamilton from C.A.R. Entertainment, and I will see you next time.